Again, my name is Nathan Peck, CEO of Cure VCP Disease and also a patient with the VCP mutation. And I'm excited for this next session. I'm honored to introduce, session 2 panel. Have Dr. Hemmo Meyer, professor, Department of Molecular Biology at, I'm going to butcher this, my German, Universität Duisburg-Essen.
Yes.
Did I kill it?
No, perfect.
Perfect. Oh, wow. All right. Then we have Dr. Peter Shen, Assistant Professor of Biochemistry at the University of Utah. And then Dr. Alyssa Johnson, Assistant Professor at Louisiana State University. And, Alyssa, I hope the hurricane, y'all are recovering from that. I'm glad you're able to join us today. So they're going to lead a discussion about mechanistic insight, including the role of VCP in protein quality control, proteasome degradation, lysosomal function, and autophagy regulation. So I want to welcome, Dr. Meyer, Dr. Shen, and Dr. Johnson. And Hemmo, I'll let you take it from here.
Thank you, Nathan, for the introduction. Peter, Alyssa, and I thought that I would do a short general introduction to avoid redundancy between the talks. So you've heard some of this already from Virginia. I'm repeating that and putting into this context. You know that VCP, or P97 as we call it, the protein, is a very abundant protein. It makes up a lot of cellular mass. It's one of the most abundant proteins in the cell, actually, and is important for a lot of different cell stress responses and also housekeeping functions. On the protein level, it's a homohexamer, which explains that mutations can actually be dominant, and it's a so-called AAA ATPase with two such ATPase domains, the D1 domain and the D2 domain, that are organized as two stacked rings on top of each other, forming an hourglass structure with a central channel with a D1 pore and a D2 pore, and also an endomain, a regulatory endomain at the periphery of this hexamer. So AAA ATPases can unfold proteins, so they can convert the energetically perfect or favorable structure of a protein back into a single peptide strand, and they need to use energy for that and hydrolyze ATP. So P97 unfolds proteins, but how are proteins targeted to P97? So the most known pathway is actually the ubiquitin-dependent unfolding or targeting pathway. So you know that proteins can be modified with the small protein modifier ubiquitin through a cascade of enzymes, and this ubiquitin is then extended to a chain. P97 can actually recognize this chain, it can recognize its substrate through this chain, but not directly, but through a specific adapter protein, the UFD1 NPL4 heterodimer that needs to bind to P97, bind the substrate, links it together, and then apparently also stick the substrate into the central pore of P97, where it's then threaded through the central pore and unfolds the substrate. So we identified this UFD1 NPL4 adapter, but most recent data now suggests actually that these substrates are actually grabbed by the ubiquitin, and ubiquitin is the unfolding tag, and it's the first that is actually translocated through this pore. The protein is then ejected at the rear side of the D2 pore of the protein of the P97, and in most cases, but not all cases, delivered to the proteasome, where the protein, the substrate protein, is then degraded. So why is P97 required if the proteasome itself has actually AAA ATPases? The idea is that P97 can actually extract proteins from cellular structures, chromatin membranes, but it also can unfold tightly folded proteins to provide at least an unfolded tag that the proteasome requires. So this is one, or the most known targeting pathway, but actually the first targeting pathway that was suggested is actually a ubiquitin-independent pathway. And we were only recently able to nail this down and prove it and show this in detail and directly. So what we showed is that P97 can unfold a PP1, protein phosphatase associated protein inhibitor 3, grab that one and strip this inhibitor 3 off the PP1 and unfold this complex or disassemble the complex to activate the protein phosphatase 1. To target this complex to P97, it requires a specific adapter, or rather a type of adapter, which we call septomain adapters, because this is a defining feature, the septomain of these adapters. In this case, I showed P37. So that binds to this complex and targets inhibitor 3 to P97 for unfolding. So you see that the targeting of substrates can be very different for P97, but the actual unfolding reaction is believed to be identical in both pathways. So the substrate is threaded through the central pore and thereby unfolds substrates, and in this case actually strips it off of its partner. The driving force is ATP hydrolysis in D1 for the major part. So basically this is the driving force. The activity of D1 is also involved, but it's not essential actually. And that's really interesting and important because it is actually most of the disease-associated mutations are actually at the interface between the N and the D1 domains. And this constitutes a communication pathway between the two domains, and it will be important to understand what the function of D1 is and the interaction with the N domain. Okay, so I mentioned already two binding factors of P97, UF1 and P47. There's actually many more of interaction partners. We call them P97 cofactors, and they are defined by dedicated interaction domains to P97. They can be distributed in different locations in the cell. They can have different functions, substrate adapters, as I mentioned, additional regulators, targeting factors to different compartments, and substrate proteins. And we will hear more about it from Alyssa, I believe. In terms of functions, so the best known function is actually ER-associated degradation, where P97 extracts proteins, unfolded proteins, and ubiquitinated proteins from the ER membrane to provide them to the ER. Similar pathways are probably functional in mitochondria, if you will, also ribosome, in the cytosol, also in the nucleus, where P97 essentially works in line, as I pointed out earlier, with the proteasome. It unfolds ubiquitinated substrates, provide them to the proteasome for degradation. I also mentioned the regulatory protein complex disassembly, or protein unfolding and complex disassembly. I mentioned the PP1 complex, but this actually happens also on chromatin. There's factors like the Q complex, the MCM helicase that need to be disassembled during vital processes in maintenance of the genome, DNA repair, and replication. So the third pathway I would define as lysosome associated, and there we don't know. So obviously complex disassembly is direct, but in terms of lysosome, we probably don't know what exactly is regulated. It's most likely indirect. There's a regulator that is possibly degraded or regulated, and that contributes to the function of the lysosome and also activity of autophagy. So I will talk in my talk about a link to lysophagy. Alyssa will talk about disease models and cell biology of P97. But the first talk is by Peter Shen, who will talk about the structure and very new, exciting insights of how the molecule works on the molecular level. Peter, it's your stage.
Thank you. All right, so it's really a pleasure for me to be here with all of you today and to share some of the recent work from my lab. I really appreciate Hemmo's introduction. It really nicely sets the stage for laying out the many unanswered questions that we have in the field. And one of the most burning questions that has motivated my lab, where structural biology and biochemistry lab, is how does VCP actually work to unfold proteins? And so I won't really give much of an introduction except this one slide here, many ideas which have already been covered by Hemmo. But as he mentioned, and as many of you know, there have been several high-resolution structures of VCP that have been determined over the years. And these consistently show that it assembles into these hexameric rings with the D1 and D2 motor domains in the 6-fold symmetric planar conformation. And major differences that we see from structure to structure is really dependent on the nucleotide state under which these structures have been determined. And we know that in an active-like, ATP-like state, the peripheral N-terminal adapter binding domains exist in this so-called up conformation. And in an inactive or ADP-bound state, these N domains are in what's known as a down conformation. But actually how these states correlate with substrate processing is unknown. And so consequently, many different models of how VCP unfolds proteins have been proposed over the years. And this is a nice summary from a review from Hemmo and Johannes. And for example, substrates, are they pulled through the entire central pore of the ring? Do substrates go through just one of the rings, either the D1 or D2 rings alone, or perhaps not even through the ring at all, and simply through a lever action by these flexible N-terminal domains? And so to determine how VCP actually works, what we really need is a structure in the context of substrate unfolding. And this is what our lab really has devoted our efforts in recent years. And so I'll spend just a few minutes talking about work that was published a couple of years ago now. And so in order to capture VCP bound to unfolding substrate, my lab specializes in purifying native complexes directly from their sources and imaging them with a structural method known as Cryo-EM. And these efforts were led by Ian Cooney, a graduate student in my lab. And what he did was he took budding yeast, Saccharomyces cerevisiae, our favorite model organism, and he introduced a flag epitope at the endogenous locus of this VCP binding partner known as SHP1. And this is the yeast ortholog of P47 and P37 that Hemmo alluded to earlier. And so with this flag-tagged SHP1 strain, Ian was able to perform really quick one-step co-IPs of native complexes directly from yeast cell lysates. And so there's an example of his gel here. SDS page analysis confirms that when he does these co-IPs against the flag tag, you get robust amounts of CDC48 that are recovered in the elution. And we also see a smattering of other proteins. All these other bands that you see here belong to other CDC48, other VCP adapters, and other substrates. And so what we do in the lab is we take this purified material and image them using Cryo-EM. And in the back here is an example of what a Cryovia Micrograph looks like. And each of these little speckles you see is an individual CDC48 hexamer, each of which is a bit noisy, but we can take each of these particles and average thousands of them together to get these more robust averages of particles in different poses. And with these higher resolution views, we can then back project them into 3D structures. And so that's exactly what we did with this yeast data set. And one of the greatest advantages that Cryo-EM offers is the ability to recover multiple structural states that might exist in a data set. And this was extremely relevant because it turned out that our particles adopted two major conformational states. The first, comprising about a third of our data set, was very reminiscent with what's already known, that CDC48 forms this symmetric 6-fold hexamer. And so this state wasn't so interesting to us. We didn't really learn anything new from this particular structure. In contrast, the remaining 2/3 of our particles resolved into this 3.7 angstrom structure that showed an asymmetric spiral staircase of the hexamer. And we knew we were really onto something special here because the structure, looking closely into the central pore, revealed an unfolding substrate density within the central pore, colored here in purple. So how does VCP interact with substrate in the central pore? And so through modeling of our electron density, it turns out that 5 out of the 6 subunits, colored red through light blue here, and both D1 and D2 wrap around the unfolded substrate, shown here in purple ribbon representation. And zooming further into this D2 ring, there's a network of deep grooves formed by large tryptophan residues. These are highly conserved, and these tryptophans function as molecular footholds for the side chains to fit into these grooves, and these side chains belong to the unfolding substrate. And so this is an important point because it explains how these pockets can accommodate virtually any side chain, and that protein unfolding is therefore not dependent on any particular sequence within the substrate, which is what we would expect because VCP would be needed to unfold a wide variety of proteins in a cell. And so zooming back out, we can see that this array of tryptophans is repeated throughout the central pore, really nicely accommodating the side chains of the substrates as they're being threaded through. So I mentioned 5 of the 6 subunits wrap around the substrate. What about the 6th subunit? We refer to this as subunit F, A, B, C, D, E, and F. It's also the lagging subunit, at the bottom of the hexagon. And it turns out that this 6th subunit, this lagging subunit, is completely detached from the substrate. In fact, the contact interfaces within this subunit itself is relatively weak. And using additional cryo-EM image processing, we were able to determine its structure in multiple structural states that we summarize here as F1 to F2. And piecing these states together implies a trajectory of motion in which this lagging subunit moves from the bottom to the top of the spiral staircase, where it can then bind the next exposed region of the unfolding substrate. So piecing our observations together led us to propose a translocation model that CDC48 unfolds substrates through the central pore using what we describe as a hand-over-hand mechanism. So I'll start this video. And what you'll see is that at any given point of this video, the bottommost subunit, this lagging subunit, is going to detach from the substrate, and where it can then climb back up to the next exposed part of the unfolding substrate. And this happens in a sequential manner, in what we refer to as a hand-over-hand mechanism. And this is topologically, therefore, equivalent to pulling a substrate through the ring. And so we had this model for the yeast complex. And so we naturally then asked, does the human protein use the same mechanism? And of course, in order to answer this question, we need to be studying the actual human protein. So as you can imagine, working with human cells is much more challenging than with yeast. You know, we don't have the luxury of growing a ton of material in a short period of time like we can in yeast. And so Yang Xu is the postdoc in my lab who has pioneered these efforts to begin studying VCP in tissue culture systems. And so unlike our work with yeast, where we simply pulled on a native VCP adapter, Yang adapted her co-IP strategy so that she actually purified recombinant flag tag P47, this VCP adapter. And then with this purified protein, she then added it into the lysates of HEC293 cells. And so by doping in this exogenous P47, she can then do this co-IP against the flag tag. And then anything that comes out of the lysate would presumably be binding partners to P47. And so this approach really was wildly successful. We got obviously abundant amounts of P47, which is what Yang pulled on, but very satisfyingly, we also get abundant amounts of VCP. And we also see a number of other binding partners and substrates as evidenced by all of these other bands that we see here in the gel. And so like with the yeast work, we also proceeded with cryo-EM imaging, a lawn of particles that you see here that we also then use as the basis of 3D reconstruction. And perhaps not surprisingly, our reconstruction of human VCP closely matches that of our yeast structure. As before, we observed that 5 out of the 6 subunits wrap around the unfolding substrate, and the 6th subunit is disengaged as if transitioning from the bottom to the top of the complex. One point of emphasis is that the P47 density, particularly for its C-terminal UBX domain, was reasonably well resolved, and that it appears that all 6 of the VCP subunits in our reconstruction have this N-terminal domain in the up conformation with UBX density bound to it. And this is also consistent with previous reports that this up conformation is associated with active VCP. And so here's the full view of the segmented substrate density that we see in the central pore. This is the substrate that's being unwound by VCP. And like with what we saw in yeast, 5 subunits in D1 and 5 subunits in D2 wrap around the substrate, with pore loops forming a helical array. These interactions are also mediated by an array of deep grooves created by side chains from neighboring subunits. The grooves are much deeper for D2 with the tryptophans compared to D1, but both D1 and D2 have grooves that can accommodate the side chains of the unfolding substrates. One thing I failed to mention in our yeast work is that we also see nucleotide density in our reconstructions for both the yeast and human structures, where the first 4 subunits in both of our structures are bound by ATP, and then the 5th subunit is bound by ADP, therefore suggesting that nucleotide hydrolysis happens towards the bottom of the spiral staircase. And conceptually, this makes sense because at the bottom, this is where, after nucleotide hydrolysis occurs, this is what promotes the disengagement of the subunit from the substrate and weakens the interface with neighboring VCP subunits, allowing that lagging subunit to detach from the rest of the complex, where it can then climb back up to the top of the hexamer. Now, one distinguishing feature in our human reconstruction is the presence of additional density above the central pore. This density is best modeled as a trimer of the P47 subdomain, and this also appears connected to the substrate density. And this suggests that P47 feeds substrates into the central pore through this subdomain that Hemmo also briefly alluded to earlier. And this position is also reminiscent of what we know about how MPL4 feeds substrates into the central pore as well, where MPL4 is also positioned directly above the opening of the VCP pore. And we see something very similar here with P47. So VCP belongs to a family of enzymes known as AAA ATPases, and many other substrate-bound structures have also been determined in recent years. And so we took 14 of these structures and overlaid the substrate and surrounding pore loop models against each other. And very satisfyingly, we found that they overlap quite well, with the 5 pore loops forming a helical array around the substrate. And in contrast, comparing all of these different models together, the position of the 6th subunit, this disengaged lagging subunit, the position of the pore loop was all over the place. And again, this is consistent that this is the subunit that needs to be transitioning from the bottom to the top of the complex. So taking these observations together about what we know of what we've observed with VCP and CDC48 and what we know of other AAA ATPases allows us to put together a translocation model. So I'll go ahead and play this animation. It's created by Janet Iwasa in our department, similar to the model animation video that I played earlier. And this, again, showing this hand-over-hand mechanism of substrate translocation in which adjacent subunits climb in a sequential manner to pull substrates through the central pore. And so in summary, I hope that I've convinced you today that VCP unfolds proteins by pulling them through the central pore using a sequential hand-over-hand mechanism, and that this process is broadly conserved from yeast to human. And looking ahead, I would love to know how disease mutations affect this translocation mechanism, as well as how different adapters influence substrate interactions. And I'll conclude by just showing 2 really quick slides about our ongoing efforts in this regard. First with disease mutations, and second with what we learned about substrate interactions. And so as what's been mentioned several times throughout this conference now, as many of you know, most of the documented mutations that cause disease are clustered within a very narrow region across the whole gene, across the whole protein, generally sequestered within the internal domain or close to the D1 ring. And we've come across this R155H mutation several times now. And so Yang in my lab developed an in vitro system where she purified recombinant wild type and R155H versions of VCP. And in our in vitro assays, we recapitulate very well-known results that this mutation causes hyperactive ATPase activity. And our structures of wild type and mutant VCP gives us an apples to apples comparison, showing that at steady state with wild type protein, in the absence of substrate and nucleotide, the VCP is stabilized in a down or repressed conformation, compared to the R155H mutation, where at steady state, these hexamers appear to be in a constitutively active conformation. And then this is my last data slide here, just showing that we've also done mass spec proteomics of our co-IP material. We've been interested to know, what does P47, what type of substrates and perhaps other adapters co-purify with P47. I'm sorry if this doesn't show up very well. It's a little bit small on my screen, but you know, a lot of these proteins I've never heard before, but very satisfyingly and interesting to me was the enrichment of ubiquitin in our P47 pull downs. It's a lot of, I guess, haziness in the literature as to whether P47 interacts with monoubiquitin, polyubiquitin, or ubiquitin at all. And when we probe for ubiquitin specifically in our co-IPs, we do see an enrichment of it, as well as other known VCP adapters, UBXN1, PLAA, UBXN7, and potentially other specific substrates that, you know, warrant future investigation. And so with that, I think it's important to acknowledge really the people in the trenches doing this work. Yang is the postdoc that led our efforts in studying human VCP. Ian did the yeast work. Our structural and AAA TPS collaborators, Han and Chris, and JC Price at BYU for the proteomics help. And a picture of my lab, funding sources, and shameless plug that we are hiring at all positions. So with that, thank you for your attention.
Yep.
So
Go ahead. I think you.
I just wanted to say that there's time for discussions now, as we agreed, right? For Peter, any questions for Peter?
All right. So I see Donna has a question in the chat. Can you comment on, for inhibitors, how many subunits would they have to be occupied for effective inhibition? So we don't, in my lab, we haven't really explored inhibitors in depth. It's a direction that we're really interested in, but we haven't done any biochemical or structural work with inhibitors like CB5083 or the NMS compounds. It's a fascinating question, very important, but this is a logical next step that we are interested in taking. All right, so Michelle asks, how do I estimate the up and down equilibrium states, how the up and down equilibrium relates to the staircase? Yeah, this is a really important question. Let me find the right slide for this. I don't really have a slide that speaks to it, Michelle, except just referring back to our reconstructions here. So with cryo-EM image processing, what we can do, I assume I'm still sharing my screen. Hopefully you guys can see this.
You're good. You're good. Keep going.
Perfect. Thank you, Nathan. With cryo-EM image processing, we can do what's referred to as image classification. And the neat thing is that the downstate of VCP is actually very stable. It forms extensive contacts with neighboring elements within the D1 ring. Whereas VCP in the upstate, the N-terminal domains are kind of just floating in solution and not very well resolved. And so if we did have N-terminal domains in the down position, then we would have expected to see high resolution features in that case. But but quite the contrary, we don't see that at all. That through our best classification efforts, all of the N-terminal domains that we see in our substrate bound structures are in the up conformation. And we can say that with a high degree of confidence. Chad, what do your VCP models suggest about the importance of the D1 loop residues in substrate translocation? This is a really important question, and I'm glad you asked it because it wasn't part of my original presentation. Let me see if I have a hidden slide for that. Yeah, so I did gloss over really briefly. This is a snippet from our yeast work where the grooves formed within the D1 ring are pretty shallow. And in fact, this shallow nature is conserved among CDC40 from yeast to human. Let me pull up the human, yeah, right here, the human model. And in fact, Teru Ogawa, I'm going to butcher the last name, Ogeru, I think.
Oguru.
Thank you, Oguru, in Japan.
Ogura.
Thank you.
Ogura.
Showed that if you mutate the D1 pore loops to be more canonical with other AAA ATPases, this leads to a lethal phenotype in their yeast system, suggesting that actually the D1 loop actually really needs these shallow grooves. And then multiple groups have shown that through in vitro biochemical assays that the ATPase activity of D1 is expendable for substrate translocation, whereas D2 is absolutely necessary. So it does appear that there are distinct features between D1 and D2. My pet theory is that the motor in D1 does not need to be as strong as D2, because all you need D1 to do is to funnel substrates directly into D2. And D2 is really what provides the strong driving force, the strong translocation motion for the unfolding substrate. So as long as you have substrate that feeds into D1, then D2 can do the rest of the work. But I don't have any hard evidence to back that up, except for all piecing together all the evidence from other papers.
Okay, I think we have more time later on after. Yeah, so we made quite some progress and the field and understanding P97 function. What I would like to focus on today is actually a role that we find really exciting in what we call, or what is called, lysophagy, so autophagy of damaged lysosomes. So a defining feature of aging cells and degenerative cells is that they accumulate cellular waste. And there's two types of systems in the cell that remove cellular waste. One is the proteasome that degrades unfolded proteins, single proteins, that are provided to the proteasome, as I explained earlier, through ubiquitination targeted to the proteasome and then degraded. And we learned over the years that VCP is really important for that. And this is the UFT1 NPL4 adapter that then binds to the substrate and helps degrade proteins, unfolded proteins that are prone to aggregation in the proteasome. There's another very important degradative system, which is the lysosome, and that degrades anything that is taken up from the plasma membrane, obviously, things endocyte host material, and this is degraded in the lysosome. And then very importantly, also anything that is funneled into autophagy, so the degradation of material of the cell that is taken up by autophagy, put into autophagosomes, and then delivered to the proteasome. And this can be proteins that are already aggregated and cannot be substrate of the proteasome, so this is sort of a backup system, but also larger structures, including whole organelles like lysosomes or mitochondria. So we know that P97 is somehow involved in autophagy is for many years. Chris Weier's labs and Paul Taylor's lab found links to autophagy. And there's actually a quite interesting recent paper by the Rubinstein lab that links P97 to a regulator of autophagy, bacillin. So P97 regulates apparently bacillin, and that could explain some of the autophagy phenotypes. We at the time took actually a different approach. We were looking at, in an unbiased way, for adapter proteins that are somehow affected by disease-associated mutations. And what we found, also a bit serendipitously, is adapter proteins that form a complex with VCP that seem alternative to the UFT1 NPL4 complex that included a protein called PLAA and UBXT1. And PLAA is really interesting because it's been linked to actually the sorting along the endocytic pathway in yeast already, the yeast hallmark. And there's actually mutations in PLA found being associated to an infantile endocephalopathy that also somehow affects the endolysomal sorting. So we became really interesting in this. And what we did actually recently was that we thought that we hypothesized that this complex could actually be involved in a stress response in this whole lysosomal system or endolysomal system. And the most dramatic stress of the lysosome is actually rupture of lysosomes. So lysosomes can actually leak, can become permeabilized, and this is really deleterious for the cell because they release all kinds of components that lead to cell death and inflammation. I don't want to go into detail here. Also, obviously, the lack of lysosome function will also lead to a lack of degradation capability of the cell. So this is very, very deleterious to the cell. Why do lysosomes actually rupture? This can be exogenous factors like drugs, lysosomotropic drugs, detergents, crystals, and obviously pathogens that can damage the membrane. And there's a lot of large body of work on that. Interestingly, can also be caused by endogenous factors, and I find most interesting is that this is linked to oxidative stress, also unbalanced lipid composition. So I'm thinking of Virginia's mentioning the ceramide link. So and also neurotoxic aggregates. So anything that is linked to aging cells and degenerating cells. So as I said, this is a major problem for the cell, and there's pathways that deal with this with these damaged lysosomes. One is actually repair that turned out to be mediated by escort proteins, and there's also signaling involved that induce reconstitution of new lysosomes. But what we focused on is actually the removal of the damaged lysosomes by autophagy, termed lysophagy. So this is actually initiated by the influx of cytosolic lectins, so-called galectins, that then get into the lumen, but then bind to the sugars and then are trapped in these lysosomes. This is also a great reporter for damaged lysosomes, because galectins can only be found in the lumen of lysosomes when they are damaged. So we damage cells with a drug called LOMA, and what we find when we treat these cells, we find a large amount of lysosomes that are actually galectin positive, right? The next step in the pathway is then actually the ubiquitination that is also triggered apparently by the galectins, and that leads to extensive ubiquitination of the damaged lysosomes. You see that here. So this is an untreated cell, and this is a treated cell. So you find ubiquitin on these lysosomes. Much of this ubiquitin is actually put on the lysosomes in order to recruit the autophagosome, the phagophore, in order to build the autophagosome and then help so that this then fuses with intact lysosomes in order to remove this whole damaged structure. What we hypothesized that this is actually linked to P97, and indeed we identified P97 or VCP, I should say, on damaged lysosomes. So when we damage lysosomes, this is undamaged cells without damage, and then we damage lysosome, we see massive accumulation of P97 on the damaged lysosomes. So is that relevant at all? And here we are looking at the clearance of damaged lysosomes by autophagy, and this is easily followed by looking at these galectin-positive damaged lysosomes. After damage, you immediately see a lot of them, and it takes about 12 hours to completely clear them from otherwise untreated cells. Right? And this clearance is absolutely dependent on P97. So P97 is involved and required for this clearance. We see that here. There's damaged lysosomes persisting over time if we knock down P97, if we inhibit P97 more acutely with P97 inhibitors. And interestingly, it's also affected by the disease mutation. And this is done by Chris Weil's lab here in mouse embryonic fibroblasts that harbor one of the disease-associated mutants. Clearance is massively delayed, and then we find also damaged lysosomes accumulating in affected muscle tissue. We think from the mechanistic point is that P97 in fact targets ubiquitinated, actually lysine 48 ubiquitinated substrates or conjugates on the lysosomes. There's lysine 63 ubiquitin on the lysosomes. That is this autophagic eat me signal, but there's also a lysine 48 conjugates, and they persist and accumulate if you inhibit P97 in various ways. So the idea is that in addition to these eat me signal ubiquitin chains, there's additional ubiquitination occurring that then is targeted by P97, and this probably removes these proteins in order to facilitate the phagosome formation. We are actually currently actively pursuing this and trying to find these ubiquitinated proteins. What are these targets, and how does P97 actually, by targeting these proteins, trigger and drive the autophagy of the damaged lysosomes? In the meantime, we actually looked at what actually ubiquitinates the proteins that are targeted by P97, and we did that obviously. So ubiquitination, as I said earlier, is mediated by a cascade of enzymes, and we actually focused on the E2 enzymes that directly modify the substrate with the help of the E3 enzymes, and there's only 40. And the readout for this, and we actually screened this by siRNA. We performed an siRNA screen, and the readout was actually microscopic. The ubiquitination of the damaged lysosomes is a very strong signal to noise. We identified a few E2 ubiquitin conjugating enzymes that affect, when depleted, affect the ubiquitination. And this is in different steps of the screen. And the last step was actually looking at whether any of these candidates are actually translocating to the damaged lysosomes, and we found one that definitely did that, and it did this massively, and not so much a related E2 that was also found in the screen, but actually didn't translocate. We then applied an assay called proximity labeling, proximity biotinylation, so to identify any proteins that are in proximity of UBE2 Qr1. This is the E2 that we identified, and we have found a really exciting and interesting set of proteins that were exclusively found in proximity after damage of the lysosome. So, and gratifyingly, this included VCP and some of the cofactors here, and obviously also lysosomal proteins and proteins associated with the endolysomal damage response. So what does UBE2 Qr1 do? It is important indeed for the ubiquitination of the damaged lysosomes, and it's actually lysin 48 ubiquitin chains that it mediates. So if we deplete it from the cell, ubiquitination is largely decreased, ubiquitination of damaged lysosomes largely decreased, and also the recruitment of P97 is therefore then inhibited, indicating that P97 is indeed targeted by ubiquitination that is mediated by UBE2 Qr1. Right? You see that here compared to control cells, and this is specifically for those that actually show a reduction of lysine 48 ubiquitin. And we think that UBE2 Qr1 and P97 actually is important for the autophagosome formation. There's a phenocopy, so we have a mild effect on P62. So this is sequestosome 1 protein that is recruited upon ubiquitination of damaged lysosomes as an autophagy receptor. This is already affected, but then there's a really strong effect on the association of LC3, which is a marker for the phagophore that forms around the damaged lysosomes. So what we, and then actually, in fact, what we see is that indeed there is a strong effect on the clearance, consistent with the effect on LC3 formation. And interestingly, there's actually also already the depletion of UBE2 Qr1 in otherwise unperturbed cells. We actually have already an effect on lysosome. This is interesting. We have an increase of the lysosomal compartment, as you see here, with a LAMP1 lysosome marker and also in Western blots. And then there is a dissociation of the mTOR kinase, indicating there is something wrong with the lysosomes, with lysosomal signaling. So indeed, there's also there's an effect on clearance and a lysosome homeostasis. Okay, so what we think is that in order to respond to damaged lysosome, one important thing is actually removal of the damaged lysosomes by autophagy and a process called lysophagy. And obviously these structures, the cargo is ubiquitinated as an eat me signal for P62 and other autophagy receptors like type 1, so other autophagy receptors. What is important though is, and really new, is that there's an additional ubiquitination, layer of ubiquitination occurring, mediated by the UBE2 Qr1 Do enzyme of ubiquitin conjugates of proteins that are then removed by VCP in order to drive lysophagy. And if this is inhibited, the clearance of damaged lysosome is impaired. So obviously the important question is, what are the targets here? And we are actively working on this. And how relevant is this really by this function of P97 in different cell types and tissues? And how does failure in lysophagy actually contribute to the disease pathogenesis? And how would one be able to intervene? I'd like to thank the people that did the work, collaboration with Chris Weil and with Christian Behrens, who did the mass spec and the screening. Thank you for your attention.
Thanks for giving me this opportunity to share our work today. So my lab is focusing really more on kind of the cellular and organismal level of VCP diseases. And we're using Drosophila as our main model system to really try to better understand some of the normal functions of VCP, and then also how some of the mutations affect those functions. So we've been primarily focusing on the last couple of years on the autophagy lysosome system because the major cellular pathology of VCP diseases are inclusion bodies and protein aggregates. And we've been using live imaging tools to really look at the dynamic process of autophagy and try to visualize what sort of dynamic processes might be going awry in when the mutants are expressed. So the major marker that we use to visualize autophagosomes is the Drosophila homolog of the LC3 protein, which is called ATG8 in flies. And then we're using to visualize the lysosomes, a lysosomal membrane protein called SPIN. And this is where we really started kind of investigating a new class of lysosomes. Because when we looked at this SPIN marker, these lysosomes were traditionally viewed as these vesicular structures, but we found that in certain cell types, including Drosophila muscles, that these lysosomes actually adopted a very different architecture. So they form these web-like tubular connections, and they're really dynamic. So we can see these lysosome tubules that can extend and retract. They can make fusions and connections with other lysosome tubules. They can bud off into discrete vesicles. So this was sort of a unique morphological feature of lysosomes that hadn't really been, well characterized in many systems. So we started by looking at, just the general properties of whether these are, actually degradative, are they functional lysosomes? So we co-imaged them with an autophagosome marker, and we found that they can take up autophagic cargo. So we see co-localization of autophagosomes within the lysosomes. If we stain them with lysotracker, we also see that they're acidic in nature. So they seem to be, in fact, degradative. So we and others have now identified these tubular structures in multiple species and tissues. And really what's been interesting is that they behave very differently in all of these different contexts. So in some contexts, like the muscle cells, these TLs are constitutively present. They don't really seem to require any stimulus. But in other states, for example, in the gut cells, we found that they are induced only in certain conditions. So, for example, starvation is one major inducer. So starvation is a major, cue for autophagy induction. If animals are well fed, their lysosomes are mostly vesicular, and then they transform into these tubular structures upon starvation. Additionally, there's some developmental cues in certain tissues that can induce them, and they can also be induced upon immune response in certain immune cells. In this case, however, these are actually not autophagic lysosomes, and they perform some specific immune functions. So this is where we started investigating VCP, because one of the first genes that we identified that is really important for the maintenance of this tubular network is VCP. So if we inhibit VCP with RNAi, we see disruption of this tubular network. If we add back the human VCP protein, we can rescue this, suggesting that this is a conserved function for the human protein as well. And so over the last couple of years, we've been really investigating two major questions. What is the normal function of VCP at these lysosomes? And really, are these lysosomes, important for the disease? So to tackle the first question, we had to find a way to tease apart VCP-specific functions at the lysosome. So this was challenging because VCP is involved in a number of cellular processes. And so we know that the major way that VCP is regulated is by binding to different cofactors that direct it to different cellular compartments or different pathways. So we identified a small cofactor called small VCP interacting protein, or SVIP, that directly recruits VCP to the lysosomes. And by manipulating SVIP, we've been able to really break the connection of VCP at the lysosomes and then really assess the importance of VCP in regulating the lysosomes. So just a little bit about SVIP. As its name suggests, it's a small protein. It's only 9 kilodaltons in size. There's not really that much to it structurally. It has two coiled regions. But the two most critical parts of the protein, there is a VCP interacting motif in this first coiled region. And then at the very end terminus, the second glycine residue is myristylated, and that allows for binding to the lysosomal membranes. So we first characterized the Drosophila SVIP homolog, which was at the time a sequence orphan, and we performed an in vitro binding assay with VCP to show that it does directly bind to VCP. If we make mutations in this VCP interacting region, we lose that interaction. And then we tagged it with GFP and looked at the localization in muscle cells, and we saw that it co-localizes with the tubular lysosomal network. And then what was interesting is that we found when we overexpress SVIP, we can actually really drive a dramatic relocalization of VCP. So when in control cells, so when SVIP is expressed at its endogenous levels, VCP is predominantly localized to the nucleus, and then you can see it faintly in the cytoplasm. When SVIP is overexpressed, we see VCP driven away from its nuclear localization and exclusively onto the tubular lysosomal network. So then we looked at the phenotypes that were associated with SVIP knockout, and we found that with loss of SVIP, we see a decrease or a disruption in the tubular architectures. And so we can kind of quantify the integrity of this network by quantifying the number of junctions per tubule. And with SVIP knockout, we see a significant decrease in the number of junctions that are observed. This phenotype can be rescued if we re-express wild-type SVIP in the muscle cells. However, if we re-express the SVIP mutant that has mutations in the VCP binding domain, this is no longer sufficient to rescue, suggesting that the direct interaction or physical interaction between SVIP and VCP is really important for maintaining the integrity of this network. Another interesting thing that we found is that if we looked and quantified the number of tubules per area, sort of a measure of the network density, in the rescue, we actually see an increase in network density compared to the wild type cells. So modulating the levels of SVIP can actually regulate the number of, the amount of tubules that are present in the cell. And as we expected, we also saw a disruption of autophagy. So we can monitor a fusion of autophagosomes with lysosomes using a dual fluorescent reporter that's tagged to this autophagosomal protein. And so GFP will be naturally quenched when it's inside the acidic environment, inside the lumen of the lysosome. So in wild-type cells, we see very little GFP fluorescence, but the M-cherry fluorescence is unaffected. However, in our SFIT knockouts, we see a significant increase in the GFP fluorescence, indicating that the autophagosomes are not efficiently fusing with the lysosomes. At the organismal level, we see defects in mobility of these flies. So on the left are wild-type flies at 3 weeks. And then on the right here, we have SFIT knockout flies. And you can see that most of them are really struggling to climb to the top of this column. This phenotype is progressive in nature. So if you look at 1-week-old animals, they really don't have much of a defect compared to the wild-type animals. But then this progresses over the course of 4 weeks. We also see a reduction in total lifespan. So we wanted to see, whether these mobility defects were attributed to muscle wasting or neurodegeneration or both. And so we looked at muscle integrity with EM analysis. So in with wild type, so this top panel is an image of the wild type muscle, and you can see you have this nice wide muscle fiber here. In the SFIT knockouts, we see that the muscle fibers are much thinner. So this is one entire muscle fiber at the same scale. And then this is another muscle fiber down here. So you can see that the muscle fibers are much smaller compared to wild type. We also saw some defects in organelle structures. So these lysosomes form these onion skin ring-like structures. And then the mitochondria really don't have a lot of their well-defined cristae structures that can be seen in the wild type muscles. We then looked at neurodegeneration. So we looked at a pair of motor neurons in the ventral nerve cord, which we stained with GFP. And in wild-type animals, these neurons are stable really throughout their life. And so you can see these cell bodies and then these dendrites that project out. In the SFIT knockouts, we see that these neurons initially develop normally. So you can see throughout different stages of pupil development that they form these pairs of dendrites. However, even starting at 1-day-old animals, you can see degeneration of these neurons. And by 30 days, there really isn't much left of these this pair of neurons. So then we wanted to ask really what is the most important tissue source for SFIP expression. So we did rescue experiments both in muscle and neurons. And we found that if we rescue in muscles, we can get a complete rescue of the lifespan. However, in neurons, we were surprised to find that not only do we not get any suppression at all, but overexpression of SFIP in neurons seems to have some toxicity. So that indicates that the tissue source of SFIP is actually really important. So just to summarize this part, we've identified the small VCP interacting protein as a cofactor that directly recruits VCP to the tubular lysosomal network. This promotes network expansion and is important for cellular and organismal health. So for the second part, we've been creating VCP disease models in flies using CRISPR. And so we have a collection now of different VCP disease models where we've knocked in specific patient mutations, replacing the endogenous VCP gene with these patient mutations. And we've been characterizing these mutations on really a molecular and cellular level. And so I'm not going to go into as much detail on the characterization of our models, but one of my graduate students, Ankita Basu, is presenting a poster in the expo, and you can visit her poster to get more information about some of these disease models. But to sort of summarize the bigger picture, we see a lot of defects that are really reminiscent of VCP diseases, and we've also uncovered some novel cellular phenotypes. Specifically, we see male-specific mitochondrial defects, suggesting that there may be some sex-specific differences related to the mutations. We also see in the A436S mutation that we see this ectopic accumulation of TBPH or TDP43 inside of the nucleolus. And then one really striking phenotype that we observed in several of the mutant lines is that there is a defect in nuclear morphology. So if we stain with a lamin antibody, we can see these concentric circles. The nuclei seem to be really dysmorphic, and they're also smaller in size. And this is really interesting to us because this is a phenotype that is really reminiscent of what's seen in patients with progeria, which is an accelerated aging disease. And so what we're using these models for now is to really try to better understand not only the disease pathogenesis, but really utilize them as a tool to screen for potential therapies. And so, we've wanted to test the hypothesis of whether or not SVIP overexpression in these disease models could suppress any of the phenotypes. And so we are working on this, and we have some preliminary data for 2 of these mutations. So the R152H mutation, so this is the R155H mutation in humans, and the C variant as well. And so if we overexpress SVIP in this mutant background, we can rescue the tubular network, and we can also see that SVIP is able to drive the mutant protein onto the TL network. And we see this similar thing for the R152H mutation as well. And we have some promising data that this also results in some improvement at the phenotypic level or at the organismal level. So this first video on the left is a climbing assay of the R152H mutations. And you can see that, a lot of them can begin climbing, but you see a lot of them really dropping to the bottom, and they can't really maintain their residence at the top of the column here. However, in the R152H mutation with SVIP overexpression, you can see them, they can move up the column faster, and then they can really maintain their presence at the top. So it seems like they have that really much more consistent strength. And we can quantify that here, and we see that really, again, for both of these mutations. So this is really promising that, SVIP might be a potential tool or molecular target that we could utilize to try to suppress some of these disease phenotypes. So just to summarize the second part, we've developed some Drosophila VCP disease models that we hope will be a valuable tool to the community to provide a system to screen potential therapies, maybe even perform some genetic modifier screens, and we've also identified that SVIP might be a good molecular target for VCP disease suppression. And so with that, I just want to thank the members in my lab. So Ankita Basu and Jordan Wall were really the 2 students that have done all of the work on the VCP disease models, our collaborators at UCSF and LSU, and our funding sources. And I'm happy to take any questions.
There's a question.
Do you know where on VCP that SVIP binds? Yeah, so it's actually right at the very end terminus. I think that it's right around amino acids 90 to 95. So we did do some mutational analysis with VCP to really map the binding site on VCP. So it's there are a couple of disease mutations that are close to that binding site, and we have actually performed some in vitro binding assays with all of the patient mutations that we've characterized so far. So there's one mutation, the P134L mutation, that completely abolishes SVIP binding, and we see a lot of similar defects with that. All of the other mutations, if they're in the N-terminus domain, we generally see reduced binding to SVIP. There's some mutations actually in the D1 domain that have increased SVIP binding, and we haven't really, explored that yet. But that's something that we're looking at, sort of the dynamics of the interaction. For the elaboration of the tubular lysosomal network, do you have an idea, or have you done any live imaging that indicates how it is formed? Is it a growth elongation of lysosomes or some kind of fusion event? If the latter, are you looking for protein mediators of that similar to mitochondrial fusion proteins? Yeah, we think that VCP is actually pretty important for the fusion of the lysosomes to each other. It's also really important for the fusion of autophagosomes to lysosomes. So when VCP is disrupted, we see defects in both of those. So we can see a lot of times tubules will still form and they'll still extend projections, but they never really make connections, stable connections to form that network. Yeah, we are doing genetic screens now to try to identify, more genes that could be involved in the fusion elongation. We know that, motor proteins are probably important. If we disrupt the microtubule cytoskeleton, that disrupts that tubular network too.
If I may ask a question, Alyssa, this is Hemmo. So basically, you tested different mutants. The current idea is that mutants are actually overactive, and this is illustrated by a higher ATPase activity, but the ATPase activity is very different for different mutations. Do you see some correlation there between increased ATPase activity and effect in your disease models?
So yeah, I believe, I think most of them have, increased ATPase activity. And, I don't know if that's, a lot of the mutants we think are probably mislocalized in the cell. So even if they have increased activity, if they're not at the right location, then that it's really not going to matter then, if they have that increased enzymatic activity. We haven't really seen any strong correlation between, mutants that have more increased ATPase activity versus more, defects in the tubular lysosomal structures. The ones, the mutations in the D1 domain are usually the most severe, and we also see those are the most severe phenotypes, with respect to the lysosomal network too.
But that doesn't really correlate to the degree of effect on ATPase activity, right? So you're basically saying more important is whether it's targeted or not, right?
Yeah, I think that, we were really surprised that, if you drive, VCP almost exclusively to the lysosomes, that there aren't defects, because it's not at the ER, it's not in the nucleus, you're pulling it away from all of its other normal functions. So we really don't know, why that is, if there's, really what sort of, compensatory.
Really exciting. You have two more questions in the chat.
Okay. Does the tubular lysosomal structures preserve in fixed tissue? So no, it doesn't. And actually, I think that's, when we started, that was, one of the reasons that I think that we were able to visualize these. So if we fix cells, we've tried a whole bunch of different fixatives, and we haven't really seen it preserved in any of those conditions. In muscle tissues, we've looked in C2, C12 myoblast cells. And so we do see tubular lysosomes there, suggesting that might be, conserved in human muscles. What is the human ortholog of SVIP? So human ortholog is SVIP. It was actually discovered in human cells first, and then it was a sequence orphan in Drosophila, so we just named it the same thing. Are there diseases associated with mutations in SVIP? So we worked with Bruce Miller at UCSF, and they screened their patient database for mutations. And we did, and I didn't show this, but we did find one patient that had a mutation, the very last residue in SVIP, which we didn't think was really going to do very much. But when we created the fly model and we expressed that mutation, it was pathogenic, and it did cause disruption of the lysosomal network. So there may be other, patients with mutations in SVIP, but, I think it's, just now, kind of on the spectrum of sort of being looked at in terms of degenerative diseases.
All right. Any other questions? Okay, can y'all hear me? Okay, my video froze. All right, Hemmo, Peter, Alyssa, thank you very much. It was an awesome discussion on the mechanistic insights of VCP. Hemmo, appreciate you staying up so late for us tonight. And, you know, we just look forward. We know it's early on in learning a lot of these things, and we just look forward to your continued work, and hopefully there's some collaborations that come out of this as well. So this concludes, you know, as this session concludes, I remind you that you'll need to exit. You know, you hit the leave button to exit out, and then you'll go back, click on sessions, and we will have our next session, session 3, where we'll be talking about new tools and omics studies. So if there's nothing else at this time, we'll stay on time and go ahead and you can leave the session right now. So thank you very much. Thank you again, Hemmo, Peter, and Alyssa.